Hello students, today we are going to discuss another important feature in molecular basis of inheritance, that is Human Genome Project. We know we know that human have 46 chromosomes in each of their somatic cells, and that 46 chromosomes, all the genes clear which are necessary for to be a human being, and we have from very beginning we have a special interest on what these genes are, where they are located, particularly which characters are represented by that which particular gene and which chromosome they are located, and how they are, that means what base sequences they have. So these all are very much creating a lot of interest in us, and so this Human Genome Project came into being. It is a mega project with a high budget, and uh, and the estimated time duration was 13 years, and several countries, not only USA but several other countries, UK, France, Japan, Germany, and many other countries, all they contributed in this mega project clear. And not only it elucidated the genes which are present in human, at the same time it develops numerous computational devices for storage, analysis of data. So as a result, the science of bioinformatics also developed with the Human Genome Project. So let us see what are the basic goals for this mega project. So we can see here the basic goals are, first of all, to sequence the whole human genome clear, and find out the interlength of base pairs and all. Next is the storage of these huge information as databases. Next is the analysis and develop the conduct computer based analysis for this clear, each part clear, proper analysis and then genes are to be identified, how many genes are there clear, now what they are having, what type of base sequence they are having, what characters they are actually representing. So all these are to be identified, and then the technology clear, the transfer of this technology which has been developed during this project will be transferred to various fields of science like agriculture, industry, forestry, etc. And the last one is the ethical, to solve any ethical, legal and social issues which will raise due to this project clear to other sectors like in, so that is also important. So we have already learned that which are the goals for this Human Genome Project, and next part is very important, that is the methodologies which are used in this project here. Now there are two ways how we can proceed. One is the express sequence tag. Express sequence tag means the particularly all part of the DNA is not coded clear. There are certain parts which are coded, which are actually used for protein synthesis, and proteins are produced according to that, and certain parts are there, we know they remain silence clear. So the parts which are coded, that means which can produce the proteins, if we go on analyze the proteins, the amino acids which are there in the protein, and according to that we can find out the codon in the RNA, mRNA, and therefrom we can also find out the DNA, respective DNA. So that is one of the way to go from express sequence tag or ESP, that is the parts which are expressed as proteins can be tagged, and that can therefrom we can find out the DNA content or DNA sequences. Now this is not very in use clear, but the method which is extremely used in this project were sequence annihilation. So sequence annihilation, what is sequence annihilation? While the genome coding and non-coding, all the part randomly are sequenced, clear, and at first, and then they are assigned to different areas or different chromosomes. So randomly coding, non-coding, all parts are sequenced, and then they are assigned to various parts or various chromosomes, clear. So that is another way for sequence annihilation. So here we are going to describe sequence annihilation as because this is the actual methodology which were used extensively in Human Genome Project. So what are the steps for sequence annihilation? So there are two methodologies you can see, explicit sequence tag and sequence annihilation. We were going to discuss this part, sequence annihilation, as because that has been extensively used. Now what are the steps? First of all, the cell, DNA of the whole cell will be isolated. So whatever in the cell, whatever the DNA is, it is totally isolated from the cell, clear? Then it is broken down into random fragments, clear, here into random fragments where we can use the enzymes like endonuclease and all, and it can be separated, broken down into random fragments. Next is after this formation of the random fragments, certain vectors are used, like bacteria. Here vectors are used which are bacterial artificial chromosome or yeast artificial chromosome. This is a prokaryotic bacterial artificial chromosome, then yeast artificial chromosome which is an eukaryotic one, and the host used were the bacteria and yeast. And in this host, these genes or these fragments which are being made by random, clear, these fragments are cloned. That means multiple copies where these fragments are produced in this host. Now another method can be used for this production of multiple copies, that is the amplification by PCR, polymerase chain reaction, can also be used. Now these fragments were sequenced by automated DNA sequencer which were generated by Frederick Sanger, clear, this and he got Nobel Prize for this DNA sequencer for forming develop the principle for these sequences, clear, how these automated DNA sequences will act. And so these sequences which were already we got from this automated DNA sequencer, these sequences are then arranged with the help of there are some overlapping fragments. Suppose they were in the one end of one fragment we got that AAA, on the other fragment also the end will have AAA, and that overlapping fragments will be placed one after one over the other, and thus we can make a overlapping. By the overlapping fragments we can arrange them in a proper alignment, and finally these arranged DNA sequences, clear, which were arranged in proper alignment, will be annotated and assigned to various chromosomes. We have 22 autosomes, we have X chromosome and Y chromosome, where they will be assigned accordingly. And these assignments were done on the basis of the polymorphism, restriction endonuclease, then the microsatellite, repetitive sequences. So these are what we got is human genome contains 3164.7 million nucleotide bases, and average gene is of length of 3000 bases, which of which the lengthiest one is the dystrophin gene, which about 2.4 million bases, which contains about 2.4 million bases. So dystrophin gene is the longest gene, containing about 2.4 million bases. Now the total gene in human is estimated to about 30,000, of which 99.9% of the nucleotide bases are more or less same in all individuals, and of which about 50% of the genes, the functions are discovered till now. And another important thing is that less than 2% of this genome codes for proteins, clear. So you can think a large part which does not codes for protein, and human genome also contains a large part which are having repeated sequences. Repeated sequences, these repeat sequence, we don't have any direct link of this repeat sequence with the protein synthesis or the functioning of the gene, but they have a quite important role in the chromosome structure, in evolution, in in this dynamics, clear. So they have a vital role in this context, but in direct protein synthesis or in direct enzyme formation, they don't have such important role. And chromosome number 1, which is there in the human, it contains the maximum number of genes, which is about 2968, and the Y chromosome, which is present in the males, contains least number of genes, that is 231 only. And 1.4 million locations are being observed, clear, where there is single base DNA differences, that is called SNPs, or single nucleotide polymorphism, clear. So 1.4 million locations are there where there are single base DNA differences, clear, single base DNA sequence differences, and that those are called SNPs, or single nucleotide polymorphism, as they exist in various forms, clear. That particular nucleotide sequence, clear, exists in various forms in various individuals, so they are called single nucleotide polymorphism. So till now we know all this, and but it is not the end. There are many various future challenges, clear, which can be met up by this Human Genome Project. Like, we till now have to find out the sequences which are there in the transposons, that is the jumping genes, the junk DNAs, ALUs, clear. So, and then the genes which are producing various diseases like cardiovascular diseases, endocrine related diseases like diabetes and all, neurological diseases. So what are the sequences, what changes are there in these particular genes which are causing these diseases can be find out. Then if we know come to know the cancer cell, there can be a sequence of the cancer cells, so there can be a probability to revert these cancer cells into normal cells mechanism. And there also this further sequencing can throw lights that how protein is acting, how tissues are forming, the organs are forming. So, and finally, not the least, we can produce the designer diet, designer drugs according to particular individual, clear. So all these are the future challenges of the Human Genome Project which are yet to be elucidated.